Right, question 2. Here's a red blood cell and 4 marks for 2 adaptations and an explanation. Okay. So just to help with the technique for this, we definitely want the description and then the function that links to that description. So you need to have these linked together. So you can't have random descriptions and random explanations. And yeah, go for the obvious stuff first. Biconcave disc. Biconcave disc, which means it's concave on both sides. You know, don't say just disc shaped or donut shaped or whatever. And there's some possible explanations for how this helps. The simplest is it increases its surface area for diffusion. Increases surface area for diffusion. You could also say there that this helps it bend and fold. It's got slightly weaker in the middle, yeah, fit through capillaries. And I'll go for something else that everyone knows about them. Is no nucleus, and so that gives it more space for hemoglobin, or more volume for hemoglobin. So it can carry more oxygen. So other things you could possibly say, you could just say it has hemoglobin, which allows attachment of oxygen instead of carrying oxygen. But you can't actually really see that from that diagram. Here's a graph with a oxygen dissociation curve. Adult normal hemoglobin, and this partial pressure of oxygen, which equals the concentration of oxygen, and this is saturation of hemoglobin with oxygen, which is how much oxygen is attached to hemoglobin. And this is the S-shaped or sigmoidal, ends up near 100% saturation, down to 0, and steeper in the middle. The normal lung partial pressure is 10, and at this partial pressure hemoglobin is 99% saturated with oxygen. Is that true? So at 10 is 99%. Let's just double check that is true. There we go, 99%. Yeah, we'll agree with that. And what would be the effect of reducing the partial pressure of the oxygen to 8? Let's try that. So let's try 8. Oh, right. No effect at all. Still 99%. Still reads off at 99%. This line hasn't gone down, has it? So no change. Or words to that effect. It maintains 99%. That's still 99% saturation. Use your answer to part 1 to explain why this would be advantageous to an organism, and it's really so that oxygen could dip a little bit and you could still get full saturation. So you can carry nearly as much oxygen despite a drop in oxygen availability, even sort of down to 6 possibly. is still nearly saturated. So blood can carry as much oxygen despite a small drop in available oxygen. The blood oxygenated in the lungs passes to the tissues. What would be the most likely cause of reduction of partial pressure of oxygen in the tissues? It's a long question for a very simple answer. What uses up oxygen? And that is, aerobic respiration, in moving muscles and that sort of thing. So the MARC scheme says exercise or respiration rate, but I would always go for, not that I can spell it, aerobic respiration. The part of the curve labelled B is much steeper. Explain how this would be advantageous to an organism. So we need the advantage. We need to say why it's advantageous. Which bit are they talking about? They're talking about this bit here. So this bit here, a small change in this axis will give a large change in this axis. So a small change in partial pressure of oxygen gives a large change in the saturation of hemoglobin. So we drop a bit of oxygen concentration, we lose oxygen from hemoglobin, which is exactly what we want, because we want it to go to the muscles. That's what that last answer led us to. So the phrase that you really want to remember is a small change in partial pressure of oxygen gives a large change in percentage saturation of hemoglobin with oxygen. So that's kind of the full thing. So the advantage is more oxygen can be unloaded. So more oxygen can be unloaded for that aerobic respiration. Llama hemoglobin has a higher affinity for oxygen when compared to human. Draw the curve for llamas and label it L. Okay, so I remember on my diagram llamas, lugworms, little babies all begin with L, and L is for left. So we're going to have it going to the left here. So I'm going to draw it. I'm going to draw it in green, and you just draw it in black. So this will start off on the same line at 100, but will be further to the left. Doesn't really matter how far. It just goes, do you know that it's going to be on the left? And it needs to still go through the origin, because if there's no oxygen there, it's going to have no saturation. And we're going to label it L. So it can't be above 100, it needs to go through 0, same sort of shape but just to the left. Incidentally, if this graph was being marked by the examiner, they would have to drop a mark themselves because they haven't labelled the origin. But you would always label the origin. So that's the curve for llama hemoglobin. Why is it necessary to have this higher oxygen affinity? Well, you should know where llamas live. They live at higher altitude with less available oxygen. So live at higher altitude with less available oxygen. So they can they can still load at this lower available oxygen. What is meant by the chloride shift in the carriage of CO2? So chloride shift. Well, you're going to get a mark for telling us what happens to the chloride and where does it shift to, and in what direction. So that's going to give you 1 mark, and then you need a little bit of reason why that would be the case. So it's not as difficult as it first seems. So let's just remind ourselves what's going on in red blood cells. We get the CO2 from respiration, from aerobic respiration. That combines with water, and that gives us H2CO3 carbonic acid, and that will dissociate into H plus and our hydrogen carbonate ion, or a bicarbonate ion, HCO3 minus. So that's the chemistry that you know, and it's this will go into the plasma and it will combine with sodium to give a sodium hydrogen carbonate, and then that's how that'll travel around. So the problem with that is that in this red blood cell that we've got, if that's moving out, then we need something to move in to balance that minus exiting, and so that's our chloride ion coming into the red blood cell. So we need to say movement of chloride into the red blood cell. That's the basic thing that you should say, movement of a chloride ion into the red blood cell. And this maintains this electrochemical neutrality. This maintains electrochemical neutrality. But they'd also give you a mark for knowing this chemistry as well.